as we dig down into the laws that govern our universe, laws dictating how celestial bodies move, how objects interact with each other, and narrow in on what can be considered absolute and what is most certainly relative, we must also take a step back from these revelations. To advance further, it becomes critical to understand the core of our own beings. A focus on the study of life itself has given us the opportunity to witness things like cloning, surgical robots, and artificial hearts. It took many hundreds of years to get where we are now. So where did it all start? One can accredit the beginning to the discovery of the cell. This occurred in 1655, almost 400 years ago. Over these 400 years, the world went from knowing nothing about our makeup to developing cell theory, the idea that all living things are made up of microscopic cells, to being able to edit our very DNA, and to finally today, where it is possible to cure diseases by the use of stem cells, cells that are not yet assigned a purpose in the body. Okay, let's look at the beginning. Imagine you yourself are Robert Hooke, the person who coined the term cell. You grew up in the 1600s and are a jack of all trades, dabbling in the laws of gravity, spring forces, and biology. You tend to keep to yourself, pick fights with Isaac Newton, and fall in love with your niece who works as your housekeeper. Okay, maybe not that part. In 1655, you turn your sights to microscopes. Here are some of the microscopes used in your time. Not nearly as fancy as the ones we have today. They were constructed using a single convex lens and a pin to keep the samples on. You, being the overdramatic scientist you are, decide, why not add a second lens to help see things closer? Under this double or compound microscope, you place a razor-thin piece of cork and let your eyes do the rest. Your observation: strict borders that made up many, many little boxes. These boxes you describe as cells, not because of jail cells, but because the little isolated boxes remind you of the living quarters of monks. These boxes, which made up the cork, were the very beginning of cell theory. The idea that all of us, any living thing, is made up of living, operating cells. Hooke's observations stood the test of time, and because of him, cell theory was refined over the next 200 years. Cell theory was developed fully in 1838 by a pair of fellows called Theodor Schwann and Matthias Schleiden. It stated that 1. Cells are what make up all living creatures, plants and bacteria included. 2. A cell is an individual living unit and a building block for everything else. And 3. Cells must come from other cells. A waterfall of studies followed this development. Technology became so advanced that in 2003, after 13 years of tedious, intensive lab work, the Human Genome Project came to its completion. This raises two questions. What is a genome, and what is the Human Genome Project? Firstly, a genome is the set of all our genetic instructions. It is our DNA, compiled and poised to program some cells to enable vision or to cause nail growth, or to heal injuries or basically any human function. Secondly, and simply put, the goal of the Human Genome Project was to identify and map every gene to determine their function. Mind you, there are around 25,000 genes that needed to be identified. Without the former scientists like Hooke and Schwann and Schleiden, this would not have been possible. This brings us to today. Today, because of these former discoveries and years of labor, it is possible to employ the research in the medical field. As an example of modern healthcare tech, we have Army veteran Ron Pastino. He was a first responder on scene at 9/11. He worked for 7 months doing search and rescue and cleaning debris. Because of this, he developed an inflammatory lung disease. He struggled with this disease for 20 years before he underwent stem cell therapy. Remember, stem cells are cells that have not yet been assigned a job. They are what human embryos are made up of, and as time goes on, they develop into specialized cells that make up proper body parts. These cells are unassigned a purpose and ripe for training. When they are injected in someone with a disease, they can be triggered to specialize their function, to become a specific type of cell, whatever kind the body needs, and attack the deficit. They send signals to the rest of the body ordering it to take a new approach and supply ailing organs with the chemicals and nutrients essential for repair. These cells are essentially an outside perspective for the body, forcing it to reevaluate its current healthcare tactics. Ron says that this type of treatment is the silver bullet to his disability.
I feel I'm 95% healed. This should fully eliminate it. I will validate it with a CAT scan in a few months, and I'll prove to the world that we have a full reversal in 9/11 lung disease.
As he waits for the time to come for his CAT scan to verify the fact that his disease has gone into remission, Mr. Pastino continues to feel at 100%. This is where we are at today. Research continues to advance our abilities to heal different diseases. There are many scientists to thank other than the three we have mentioned here, but this provides a neat and simple picture of how our technological advancements have grown from being able to view cork through a microscope to curing lifelong ailments. Let this serve as our reminder to take a step back and break things down into their components, their building blocks, as we go from understanding ourselves to understanding how we interact with the world around us.